She is a research scientist at Merck Sharp and Dohme, where she designs novel and greener processes to manufacture drugs and pharmaceutical intermediates. In her role, she works with teams of biologists, chemists, and computational scientists to harness the power of biology in organic synthesis. Before coming to the US, Dr. Fryszkowska worked at Dr. Reddy's Laboratories in Cambridge, UK, and at the University of Manchester, where she applied biocatalysis in chiral synthesis. With over 10 years of research in enzymatic reactions, she is passionate to combine the worlds of chemistry and synthetic biology to transform the chemical and pharmaceutical industries. So thank you so much. There are a few chairs right over here, if you don't mind.
And there's a cable.
Yeah, mine is right under a cord.
It's like a limbo.
Yeah. Back there. Take this seat
So thank you very much, Ellen and Alison, for having me here, and thank you for choosing to be here on such a lovely spring evening to listen about color. The title of my talk is Chemistry and Biology of Color, and it was inspired by my conversation with Alison, who said that there are so many artists and maybe people who are not necessarily scientists interested in synthetic biology, interested in chemistry, and it would be really cool if somebody could tell about the link between the biology and color, because we live in a very colorful world, and this color comes from somewhere. So before I start, I actually wanted to ask how many of you are scientists? So who is a chemist here? So I'll be the only one who's a biologist of some sort.
Some sort.
And who's not a scientist, like just passionate about color. Okay, so I try to prepare my talk so it is a mix of science and non-science, interesting facts. So don't get discouraged if you get lost in the nitty-gritty of the structures I will be showing to you. But hopefully you'll enjoy the next 45 minutes or so, and you will appreciate the link between chemistry and biology and color. So Alison gave a short introduction to what I do, and let me translate it to what it means in practice. So this is just an intro slide. I'm originally from Poland, so hence my weird accent. And then the last 10 years I lived in the UK, first in Manchester and then in Cambridge. And I moved to New York about 7 months ago, where I worked for Merck Research Laboratories as a research scientist in biocatalysis. So I will explain to you what biocatalysis means in this talk. But basically, what I do on daily basis, we're trying to use the enzymes and the power of biology to make drugs better, faster, in a more environmentally friendly way. So we're using biology as a tool to make molecules that we then use as drugs. So for me, there is a strong link between biology and chemistry, although I'm a chemist by training, and there will be a lot of chemical structure, so hopefully nobody's going to have nightmares tonight dreaming of the chemistry classes. So my talk has nothing to do with what I do as my day job. As I mentioned, it was inspired by a conversation with Alison. And I noticed that in New York, everyone is just crazy about color. Like, we love rainbow bagels, there's rainbow toast, there's rainbow coffee, you can have technicolor nails. And I thought that it would be nice that we have some kind of, maybe a bit of a lighter topic to talk about. And in my talk today, I'll try to answer the question, what's color? Why do we see things as colorful? And then I will tell you 5 I call them bio stories of color. So we'll be talking about purple, blues, pink, yellow, and greens. And let me start from a question. Have you ever wondered what color is? Like, why do we see color? Why a flamingo is pink and a banana is yellow? What makes those molecules, what makes those objects colorful? And color is connected to our perception of light, so it's a phenomenon of light. And it's linked to what a human eye can detect and recognize. So what our eye receptors are capable of able to perceive. And obviously other animals or other species can, some of them cannot see color, some of them see different wavelengths of light, but what a human eye can see is a so-called visible light. So this is the light which gets the wavelength of 390 to 600 nanometers, and it's referred as white light. So white light, as we all know from the Pink Floyd cover, if you direct the beam of light on a what do you call it? Prism. Thank you. Then it will split into a rainbow, right? And this rainbow is always red, yellow, green, blue, purple. And the same rainbow we see in nature. And this is related to the wavelength. So the wavelengths can have different frequency, and this frequency goes from 400 nanometers to 700 nanometers, and we see it as a rainbow. But if you overlap all these wavelengths, we see the white light, and that's the visible spectrum visible to a human eye. So now a bit more about theory of color. So the full spectrum, white light splits into the rainbow. But we also talk about primary colors, so yellow, blue, and red. And there was an artist from Denmark, Piet Mordian, which some of you may know because he's got a couple of paintings in MoMA, who said that in his paintings he can only use those three colors. And he actually had an argument with one of his artist colleagues who wanted to use other colors than that. So he was quite extreme in what he perceived as acceptable colors. But we of course know that those colors, there's more to life than just three primary colors. And if we overlap yellow with blue, then we get green. If we overlap yellow and red, then orange. Magenta is a superimposition of blue and red. And if we superimpose all of them, then we get black. So why is banana yellow? The banana is yellow because the white light, so all this rainbow, gets absorbed by the banana skin, and the only color that is not absorbed and is reflected from banana is yellow. So that's why what a human eye can see is the yellow color. So this means that in the banana skin there must be some kind of molecule or something that catches all the rest of the spectrum and reflects only yellow color. So I like to think about it or explain it as a light catcher. So this is another example of art by Janet Ehelman, who was weaving these fishnet-like structures that catch light. So think about it this way, that the bananas get those kind of fishnets that absorb some of the light but reflects another, and this is the color that we perceive. So as I said, I will tell you stories about color, about the molecules that catch colors, and also try to show the link how nature builds those molecules. I'll be talking about biology and chemistry. And I'll try to the best of my ability tell you how we can engineer the color in E. coli, although, as I said, I'm a chemist, not a molecular biologist. So I will tell you about purple, bromo indigo, and indigo, about yellows and reds. I'll start from purple, actually from Tyrian purple. And this is a story that I heard when I went on holidays to Morocco. And as you know, purple color in our culture is associated with power and wealth. It was the Caesars of Rome that wore purple, and the royals wore purple, so hence this kind of purple is called Tyrian from the name of a Roman emperor. And why was this color so powerful? Why was it so important? And why it signified wealth? So it was because it was very expensive. So to get the purple dye in the ancient times, people were fishing for those predatory sea snails to get the purple dye that this snail produced. And to get 1.4 gram of dye, they needed 12,000 snails. So you can imagine how expensive it was. And this 1.4 gram of dye, I don't even know how much it is in ounces, sorry about it. But
28 grams an ounce.
So a tiny amount. So that was only enough to dye a trim of a robe. So now you start to see the connection between the wealth and the purple color. So this story that captured my imagination was that when Cleopatra wanted to impress Mark Antony, she ordered his sails, the sails of her fleet, to be dyed in purple color. Not only did she have them dyed in purple, but they also smelled of roses. So this was a display of wealth and power in the ancient time. So the color, the structure of this light capture that is responsible for the purple color was identified not that long ago, about 100 years ago, in Germany, and this structure is called dibromo indigo. So it's got two bromine molecules. It's quite a simple molecule. It's nothing really complex in terms of chemistry. And
can I ask a really stupid question?
Go on.
When you say a catcher, does that mean if you only have that molecule, it catches all the other colors of light and only reflects purple? Just that molecule?
So that absorbs the remaining wavelength of the...
Just that one molecule?
Yeah, and reflects that.
Oh, wow.
Yeah. So I know that dibromo sounds very scientific, but the indigo, I'm sure every one of you heard of, because this is what makes our genes blue. So the blue color was actually a little bit cheaper because people could get it from a plant. There was a plant that Indigofera tinctoria that grew in India. So indigo wasn't as expensive as a Tyrian purple color. And the structure of indigo is actually very similar to the structure of bromo indigo. So the difference is just those two bromo atoms. So actually there are three colors. So there is indigo, which is blue. There is monobromo indigo, so with one bromine atom, which is purple. And there is red purple, which is dibromo indigo. So now about the biology. So how to make bio-purple or bio-indigo. So before I get into nitty-gritty, I need to explain a couple of concepts in synthetic biology and chemistry. So when people engineer things, when we build things, we like to design robots that will do this, that will build it for us. So this is a picture of an assembly line from a car factory. And nature actually works on exactly the same principle. So nature designs their own robots that will then transform one molecule into another, and then into another, and that add another thing until we get the indigo. So this kind of natural bioassembly line we refer quite often as a biological pathway, and it looks like this. A compound A gets converted by a biorobot, which is called enzyme, to a compound B, then a compound C, and eventually we get to indigo. So what are enzymes? What are those biorobots? So the enzymes are proteins, and they have all sorts of beautiful structures. I just gave you 3 examples of proteins with a catalytical function. And basically what these proteins or enzymes do, like robots, they are trained to do one action, one particular chemical transformation, and nature aligns them in the same way as we have an assembly line, and then they convert a compound A into different molecules. In this case, it's indigo. So how do chemists think about this biological pathway? So we have something very simple at the beginning, so it can be sugar, like glucose, and then we make another molecule, we add a couple of things to another molecule, and we get another molecule. And this is where there's a little bit of disconnection between chemists and biologists, because we think like this. So a biologist thinks enzyme, 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 and a chemist thinks molecule, molecule, molecule. And sometimes we have problems communicating, but what actually biologists mean, they actually quite often mean even further up, so they think genes. So there will be a piece of DNA, which is like a blueprint for this particular enzyme. So this piece of DNA will be typically named with a three-letter code. So in this case, TRP, which is for tryptophan, and then they would have consecutive letters or numbers. So like here we've got TRPE, D, and C, because these are the enzymes or genes responsible for synthesis of tryptophan, and they are on the DNA strand. So there's a link between a DNA, which codes the enzyme, and then the enzyme has a function, and this function is to convert the compound A into B and C into D. So I hope this is not that complex, and you gather the idea how nature builds its complexity from small things to complex molecules. So how is bio-indigo made by humans? Because obviously we don't want to be reliant on plants. Even more so, we don't want to be reliant on snails. So people from 1980s started to engineer bacterial strains so they can make a bio-indigo. And in 1984, Amgen, which is a very well-known biotech company, published a patent where they patented the use of an enzyme which is called dioxygenase for the production of bio-indigo. So what this enzyme did, it converted the indole by adding 2 oxygen atoms, and then into spontaneous transformations, which is water elimination and air oxidation, you get indigo. So, ta-da! You give indole to bacteria, then it oxidizes it to this structure here, and then the magic happens, and the blue color is made. So that was quite an achievement at the time, but they wanted to improve it because they wanted to show that they can actually make this blue color from something more simple than indole, because in this particular case, they needed to feed the bacteria with indole and then it turned it into blue. So 10 years later, they published a paper in Nature about designing a biological pathway to make indigo from glucose, which involved 9 genes, so 9 enzymes, to make it from glucose. And this is, so obviously they had a starting point, so they had the actual enzyme that was turning indole into indigo. And then they improved that enzyme because it wasn't the best performing enzyme, and as you can imagine, you want to have a lot of blue color from the bacteria. So they improved this enzyme by so-called directed evolution. And they added altogether 9 genes, but I'm showing here only 6, which were the genes from tryptophan pathway. So tryptophan is a natural amino acid. We all have it in our bodies. And one of the compounds on the way to tryptophan is indole. So what they did, they hijacked this pathway and redirected the indole, so it wasn't going only to tryptophan, but it was being converted by naphthalene dioxygenase to the indigo color. So that was quite a feat to design, and they showed how to feed bacteria, in this case it was E. coli, with glucose, and how to obtain blue color.
Did they have to also give them tryptophan because they weren't making it?
No, that was from glucose, and I don't remember what the nitrogen source was, but they deregulated this enzyme, so it wasn't converting indole to tryptophan.
So I'm saying they had to supplement it with tryptophan, probably, in order to keep it alive.
I didn't read that much detail, but I know that they definitely played with that last enzyme.
Maybe they didn't completely hijack it, left it enough to keep it alive.
So obviously that was a company, but there are examples where people who are passionate about science can do it. So if you're interested, there were two teams who took part in IGEN competition. So IGEN stands for International Genetically Engineered Machine Competition, where students try to build those kind of biological pathways in a modular fashion. So it's a little bit like building Legos, but building them in bacteria. So they try to design parts that will convey a certain function or feature, and then install those parts into E. coli. So there are the links if anyone is interested in learning how to do it. So I covered blue and purple, and now we will move to flamingo pink. And that was inspired by another holiday or vacation, I should say, of mine. So I went to Mexico, and in Mexico I heard that flamingos are actually not pink. Flamingos are born white, and they get the beautiful pink color from the food they eat. So they eat a lot of algae, and as they grow, they turn pink. So the story I'm going to tell you, by the way, if you want to see them, you have to go to Yucatan, where you can see flamingos in nature. But the story I'm going to tell you will be about all sorts of colors, so reds, pinks, oranges, and yellows, because they're quite related, as you will see in a second. So those dyes, those colors that we have in tomato or carrots, or that make flamingo pink, they're used by nature to either harvest the light, so to help photosynthesis in case of plants, or they protect against the photooxidative damage. So the light can damage the cells. So we need protective molecules that will prevent the light from destroying our cells. So this is their natural function. And when we look at this structure, we have two groups. So one of them, the first group is called carotenoids, and the example of this is lycopene, which makes tomato blue, and beta-carotene or carotenes in general. Everyone heard about them. These are the molecules that make carrots orange. And there are also compounds called xanthophylls, and an example of xanthophyll is the yellow color from banana. It's called zeaxanthin, and the flamingo pink astaxanthin. And as you see, the structures of those molecules, they are quite similar. They are quite complex, but quite similar and symmetrical. And you see that carotenoids are different from xanthophylls because they have oxygen atoms in those rings. So how does nature make those complex structures? And nature likes simplicity, and nature is a little bit like an engineer. So if you look at the lycopene, which's got 40 carbon atoms, you can see that this is a symmetrical molecule, so you can chop it in half, and you get two identical fragments, which are C20 and C20. And then you can chop them again, and you get C10 and C10. And you won't be surprised because we'll be chopping again. And now we get 8 identical units of 5 carbon atoms that nature uses to build a much more complex molecule. So indeed, the basic building block for carotenoids and xanthophylls is isoprene. And then what nature does, it links 2 bonds to give C10 unit, and 10 and 5 gives 15 carbon atom unit, and 20, and then it glues together 2 20s to give C40. So when that happens, when that biological pathway happens, because at each of the stages there will be an enzyme linking those parts together, we get a compound called a phytorene, which is not actually a colorful compound. And if you look really closely, the difference between lycopene, which is red, and phytorene, which hasn't got any color, is those double bonds. So the lycopene's got double bond in those positions where phytorene doesn't. So as you might have guessed already, there will be an enzyme introducing those double bonds to turn phytorene in lycopene. So carotene will be a very similar structure, but if you interconnect this atom with this atom, you'll get that ring structure. So again, there will be an enzyme catalyzing this cyclization reaction at 2 ends of a molecule to synthesize carotene. And then the yellow color, the zeaxanthin. This is carotene plus extra oxygen on both ends. So there will be an enzyme catalyzing the oxidation at both ends of carotene to give yellow. And this is how it looks, the way biologists look at it. So arrows, 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 and then genes coding those enzymes. So in this case, because this is a carotenoid pathway, they're called CRT, and then different letters of the alphabet. So flamingo pink, I will talk about it a little bit more later. Now this is how the whole pathway looks. So it's a little bit more complex. So we have here the enzymes that introduce double bond, they're called desaturasers. We've got cyclase that changes lycopene to carotene, and we've got a hydroxylase that changes transforms carotene to leucine or zeaxanthin. So this is the pathway. This is the biological assembly line. So some other examples of yellow colors. So you will see that yellow is quite similar. So zeaxanthin is the same color in banana skin as it is in a corn, and the Latin name of corn is zea, so hence the name zeaxanthin. Then lutein, which kale and spinach are full of, is quite similar to this one. The only difference is the position of this double bond. Then vioxanthin, which is in pansies. So that one's got another atom of oxygen in the ring, in one on each side, and that what makes pansies yellow. And those gene clusters responsible for carotenoid biosynthesis are present not only in the plants, but they were found in various bacteria, in microorganisms. So now we can start mining or using the pathways that were already engineered by plants in microorganisms to engineer those colors in exactly the same fashion as I showed you. Okay, so now pink. Flamingo pink is actually the same pink as a salmon pink, is a pink in some of the crustaceans, some of the shrimps. And it's a color which FDA approved as a food pigment and additive, and not surprisingly, it's in huge demand because we love eating salmon, and if the salmon is more pink, then we think that it's more healthy or more delicious. But it might be that the salmon was supplemented with the astaxanthin. So the natural source of astaxanthin is microalgae, Haematopus pluvialis, and you can get about 40 grams of that dye from a kilo of biomass. So this is a massive improvement in comparison to the snail and purple, but not exactly the way how big companies would like to make a pink color for the salmon supplementation. So there's been a lot of work done to engineer those pathways into E. coli and other bacteria. And the recent efforts allowed to produce the astaxanthin in 90% of total carotenoids. So what you have to remember that if an organism or bacteria has that pathway, it can make a mixture of all these compounds because these are the intermediates that it makes to get the final molecule. So 90% in this case of the final astaxanthin is quite an achievement. And unsurprisingly, big companies like DSM are very interested in engineering microorganisms. In this case, they engineered yeast and fungi to make those molecules. And again, you see that the nature builds those pathways in a very additive fashion. So we're starting from carotene, and we need to add 2 oxygen atoms to each ring to make astaxanthin. And there are 2 enzymes. So one CRTW that adds the ketone oxygen, and the other one CRTZ, which adds this hydroxyl group. And as you can see, this pathway is mixed, so you can go in the same way to the same molecule, just benefiting from the modularity of of the biological pathway. If somebody is interested in the detail of this pattern, it's got like 400 pages where you will have exactly described how to do it. If you want to do it for commercial purposes, then you would have to pay royalties, but if you want to do it for fun, then that should be for free. Okay, so it's not only big companies that can build those colors, and students can do it as well. And now I'd like to tell you about a project called eChromai, which was another project from the Amgen competition. This was a project accomplished by a team from the University of Cambridge. And I will try to play a short video where they explain how they made E. coli colorful. I'm going to disconnect it for a short second and
eChromai is an experimental summary. Between designers and scientists working on synthetic biology. In 2009, 7 Cambridge University undergraduates spent the summer learning the tools of synthetic biology, which is essentially a new approach to genetic engineering. Using standardized sequences of DNA in a format that's called biobricks, they learned to engineer bacteria. They designed their own biobricks using genes copied from existing organisms, inserted them into E. coli, and created bacteria that secrete colours visible to the naked eye. eChromai went on to win the International Genetically Engineered Machine Competition at MIT in 2009.
And joining us now is one of the winners of the iGen competition. Welcome to Science Friday, Ms. Muller.
Hi, thank you so much. I'm part of the Cambridge 2009 iGen team, and our project was called eChromai. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change colors visible to the naked eye.
So let's say if you put a swab of bacteria in a polluted river, bacteria would just change color.
Yes, exactly. So you probably want to put a sample of your water on a bacterial plate, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
As designers, we worked with the team to explore eChromai's potential as they were developing it in the lab. And together we imagined a timeline proposing ways that living colour could evolve over the next century. These scenarios, some of which are shown in this film, explore the different agendas that could shape eChromai's use and in turn our everyday lives. One of the first real applications for this technology may arrive quite soon: a cheap disposable biosensor for testing groundwater contaminated by arsenic.
Bacteria could also be used to produce natural colourings and dyes. By 2015, there may be a profession of people who hunt for new pigments and the genes responsible, bringing them back for use in the food and textile industry. By 2039, you can go to the supermarket and buy the simple probiotic yogurt for cheap, personalised disease monitoring.
Okay, I'm going to stop it here because that goes a little bit too much into the future. But, but you can tell that by 2015 we do have the technology to build the color into bacteria. Actually, it's a little bit less glamorous than diving into oceans for the colors, because what we do, we just mine the databases of the organisms that have the DNA sequence, and we just look for those genes. So those explorers that they imagine, they are more of a computer. So you can be really lazy and you can do these things from your sofa. I think it gives you a little bit of a taste for how this technology is used. They actually went a step further and they linked the color production with bacteria responding to some of the pollutants. So that's what they were trying to explain, that if the bacteria can detect or scan in its environment, gets some kind of molecule, then it changes color. So another color I would like to tell you about is the egg yellow. And we learned in a previous story that yellow can be linked to carotenes, and actually carotenes are the colors that give the egg yellow the beautiful yellow hue, and it depending on what the hen was fed with. If that was the corn, then it will have zeaxanthin. If that was petals or grass, then that will be lutein. And the hens do not have those kind of plants in their feedstock, then they will produce not so yellow eggs. So we naturally want to get drawn more to bright yellow eggs.
I noticed that the egg yolks in Europe were darker orange than the egg yolks in the United States.
So they can be supplemented as well. Ruin it for you. But you can feed chicken with corn, and then it will be yellow. You can feed chicken with marigold petals, and they will be then brightly yellow. So it depends what the hen is eating, and those kind of natural colors or corn or marigold are allowed to be used as additives to what the hen eats. But it's really important for us to actually eat a lot of carotenes, because this is vitamin A, and vitamin A was one of the first vitamins discovered. It was discovered in 1990. But there's of course a vitamin B, and that was also isolated from egg, and this is called riboflavin, which is from Latin word flavus, which means yellow. But surprisingly, it was actually isolated from the egg white. So the eggs contain two types of yellows. One are carotenes, which are in the egg yolk, but also egg white is full of yellow. And this yellow dye was isolated from egg white in 1933. And its structure was determined to be this a year later. And to get 15 milligrams of that dye, they needed 1,000 kilos of liver. So it is also in the liver. So you get the idea that the biochemistry 100 years ago was a really tough job, because you had to collect a lot of mass of the biomass to get tiny quantities of material. And in 1935, in Germany, two groups synthesized riboflavin. And that was sort of a breakthrough, because they pretty much eliminated the reliance on the diets when it comes to the riboflavin to the vitamin B2. So another thing that happened around the same time was that in 1932, Warburg and Christian isolated what they called old yellow enzyme. So that was an enzyme or a protein that was yellow. And that was the first discovered flavorful protein. So flavor, again, yellow, and protein is a protein. So why was this protein yellow? So this is the structure of this protein. As you can see, right in the middle, there's this structure here. And this is exactly this riboflavin molecule inside the enzyme active site. So this molecule gives a yellow color to the protein, and those enzymes, flavoproteins, are very important in oxidative reactions, in catalyzing oxidative reactions in our bodies. Chemists like myself can use those enzymes to synthesize molecules because they reduce double bonds or introduce hydroxyl groups or oxidize in general. If someone is interested in this topic, there's a whole review about those enzymes. But this is the first example where the color gives the protein a function, so it helps it to catalyze those chemical reactions that are necessary for our body to function. And this is a link to my to the company that I work for, because initially when people were trying to get a riboflavin, they tried to isolate it from natural sources. And at Merck here in Rahway, New Jersey, Merck patented a process for the isolation of riboflavin from the biomass, from the fermentation biomass. But that wasn't a very efficient process. So in 1950s, they started working on another microorganism, bio-indigo, which is another bacteria which is a natural overproducer of riboflavin. They went back to the same project in 1974, trying to increase how much of riboflavin the strain was producing, and then they went off patent. So a German company, BASF, used the strain, engineered strain, for the production of riboflavin. This is a very competitive market, so Roche, which is another big chemical and pharmaceutical company, replaced its chemical production by a biotechnological process with another bacteria called Bacillus tips. More details, if anyone is interested in the science itself, you can find in this publication. And this is just a quick slide, not to scare you off, but to show you again that the nature likes the modularity. So basically, those arrows in yellow are the enzymes from the first strain, from Ashbia, and the ones in blue are from Bacillus. And you see that to get to the same molecule, two organisms use different enzymes in different combination, but the biological pathway in the end leads to the formation of the same molecule, in this case, riboflavin. Okay, so this is the last story, but probably the most important for the biology. So this is a story about fluorescent green. This is one of the most important colors for the human civilization. It was so important that this color got a Nobel Prize in 2008. But before I tell you why it's so important and why it got a Nobel Prize, I'm going to tell you the difference between fluorescence and luminescence and just a simple yellow. So I told you we perceive banana as yellow because it reflects the yellow color, but there are some molecules or some organisms that absorb the light and then emit the light. So this is not reflection like in case of banana, but this apple, this glowing apple absorbs the light and emits the light. And this is called fluorescence. And there's also another physical phenomenon which is called bioluminescence, and that doesn't require any light input. It's usually a chemical reaction or some kind of physical process that makes the object glow. So it doesn't require the light. Okay, so now back to the green fluorescent protein. So in 2008, these two gentlemen, Osamu Shimomura, Roger Tsien, and Martin Chalfie, all working in the US, won the Nobel Prize for the discovery and work of green fluorescent protein. And here's a photo of Osamu Shimomura holding the bottle with 5 milligrams of fluorescent protein that won him the Nobel Prize. I was particularly amazed or amused by how humble those guys are. And for example, Martin Chalfie, when asked how he learned about winning a Nobel Prize, he said, "Oh, I decided to find out what schmuck won this year. So I opened my laptop and I found out." So that was very humble of him. But let me tell you how it happened that they got this Nobel Prize. So Osamu Shimomura, who came to the US from Japan, he was fascinated by bioluminescence, and sometimes in science you need to be lucky, and he actually wasn't looking for the green fluorescent protein. He was fascinated by the bioluminescence. And initially what he actually isolated from the jellyfish, which bioluminesce, was 5 milligrams of aquamarine, which is the protein which gets blue color and is a bioluminescent protein. So to get those 5 milligrams, he needed to collect 10,000 jellyfishes. And it became his obsession. He actually got involved his family into collecting the jellyfish. For over 20 years, all their family holidays was to go to Pacific coast and to fish out the jellyfishes. And they actually developed a tool that enabled to cut this rim of the jellyfish, because that's where the color was. So he estimated every year him and his family collected 50,000 jellyfish, and he multiplied it by 19 years that they went there. So that gives a number of 850 in his charisma. And what is actually fascinating about this story was that he wasn't looking for the green fluorescent protein. He actually wasn't interested in the function or how it can be used. He just wanted to explain why jellyfish is luminescent, why it glows in the dark. But the person who actually so in the process of studying this bioluminescence, he isolated the green fluorescent protein. And the person who actually sparked the whole revolution related to green fluorescent protein was Douglas Brasher. And he is actually the biggest loser of the story, and I'll explain why. So he had this brilliant idea that you can use the green fluorescent protein as a tracer molecule and use it to report information into the protein. So he had this idea that if at the end of the gene he adds the gene of green fluorescent protein, when that protein of interest is being expressed, the cell will glow. And he will know exactly where that protein is expressed, because that part of the cell will be glowing. So he also started fishing for jellyfish, and he started collecting the jellyfish. But what he wanted to get from the jellyfish was the mRNA. So he wanted to get a piece of DNA to actually clone it. And he cloned it. He got the mRNA. He cloned it, but it did not fluoresce. And it did not fluoresce because the protein he cloned contained 25 amino acids as an intron. So intron is is a part of DNA part of genetic information that, although it's present in a gene, is not necessary for the production of the protein. So it is removed in the process of translation and transcription. And the guy who actually managed to clone the fluorescing green protein was Martin Chalfie, and he cloned that gene without these extra 25 amino acids. So that's why David Crusher was a big loser of this story, because 25 amino acids cost him a Nobel Prize. And unfortunately, there's a rule that the Nobel Prize can be given to 3 people only, and there were 4. So all of them in their speeches acknowledged the input of David Crusher because it was his idea. He was just unlucky because he ran out of funding and he was unable to carry on his research. But he was generous enough to actually give his gene to Martin Chalfie, who then figured out that they have to remove 25 amino acids from its sequence. And he published a paper in 1994 where his group cloned the green fluorescent protein into a worm, with the Latin name here. And also on that Petri dish, you see E. coli on the left-hand side. This is standard E. coli, and on the right-hand side, it's a glowing E. coli with expressed green fluorescent protein.
Mark Zimmer actually gave a talk here last year. He's the one who wrote the book that you got a lot of this from.
Yeah.
And
so
he said that he actually got a letter from the Nobel Committee, and they insisted that he come because he had written this definitive history. They interviewed him to try and figure out who to give the prize to. And so they actually invited him and his wife to the ceremony. So he had a picture of himself all dressed up in a tuxedo. It's exceedingly formal.
Yeah, so sometimes scientists get unlucky, but he had this brilliant idea, and he was a fantastic person to share it, because he cloned the gene and gave it to another lab, although himself he wasn't able to finish the work.
Didn't he quit science and end up being a cab driver?
Excuse me?
He ended up being a cab driver.
Oh, did he?
Yeah.
I thought that Roger had seen and offered him a job in his lab, and he worked as a scientist.
For a while.
He might have read a stint.
That's not winning the Nobel Prize with me yet.
Okay, so what is green fluorescent protein and why does it fluoresce? So this is a structure of fluorescent protein. It's quite simple. It's built of 238 amino acids, and it looks like a barrel made of 11 sheets. And inside, so this barrel is quite often referred as a soda can. And inside of this barrel, there is a so-called chromophore, so the molecule that is responsible for its fluorescence. And this chromophore is referred quite often as light in the can. And how is this molecule made? So it's made from 3 amino acids, which are inside of the can, and then they undergo cyclization and spontaneous dehydration and oxidation to make this. I'm afraid it doesn't show too well, but this fragment of the molecule is a fluorescent green, and this is what gives the color. So the can, the protein structure actually protects the fluorescing chromophore from the environment, so the protein glows in a lot of different under a lot of different conditions. And Roger Itzin, who got the Nobel Prize, was the person who explained why green fluorescent protein fluoresce, and he actually designed a lot of different colors. So this is indeed a bio-rainbow.
Wow.
And so one of the most striking mutants fluorescing in blue contained a different amino acid in histidine in place of tyrosine. he created mutants that fluoresce faster, brighter, better. And just to give you an idea of how subtle those changes are, so the green fluorescent proteins got here this tyrosine. So if you change this tyrosine to histidine, it's turning blue. If you change it to tryptophan, it's a light blue. If you change this amino acid, which is threonine, to glycine, it's yellow. So they created a whole spectrum of the colors. And what do you do with those colors? So the first thing scientists does Petri dish with all these colors. So this is a Petri dish with all the variants of E. coli expressing different colors. Obviously, there's a more profound use for which it got the Nobel Prize. So you can start introducing those genes into the cells. So you can introduce one gene that will enable you to distinguish the shapes of the cells and where that protein was expressed. You can introduce several different colors of green fluorescent protein, and now you start seeing the granularity of cells of the processes that happen. And green fluorescent, I think as for 2015, about 3 million experiments a year are run with green fluorescent proteins in medicine, in biology, because those proteins, when they fluoresce, help us to explain the disease, help us to explain the natural function of a lot of proteins. And here is a photo of a brain bow. So this is a mouse brain, where mouse brain was engineered in such a way that it had introduced different fluorescing proteins into it, and that helps the scientists to track the neural transmission, so how the information in brain is passed through all the neurons. And this is actually quite recent. This is an MIT team that achieved that a year or 2 years ago. So I've got another short video. I'm not sure how we are.
I think we're running pretty low.
Okay, so I'll skip the video. That was about a Kickstarter project about glowing plants. So that was one of the first synthetic biology funding campaigns on Kickstarter in 2013. They wanted to raise 65,000. They raised almost $500,000 to engineer glowing plants. I would really recommend watching this video. But what happened as a result of this campaign was that there was a lot of controversy about releasing genetically modified plants into nature and giving it away to people, because that was what they offered in the campaign. And as a result, Kickstarter changed the rules, and now it's impossible to offer genetically modified species as a reward for investing in people's projects. You can buy glowing fish if you want to, so you can look at the fishes in the dark. Green fluorescent protein also has got its own monument. It's a sculpture by a sculptor Julian Gos Andrade. This is at Friday Harbor Laboratories, where Osamu Shimomura went on his holidays to fish out the jellyfish. So you have to go to Pacific Coast to actually see it. If somebody is interested in getting those slides, so you can have references to all those papers that I cited in my talk, then please drop me an email, and I'm happy to forward you the slides. There are also a couple of books about illuminating the disease and biological stains and colors that may give you more information about how the biocolor works. So that's all.
you said nature likes modularity. What do you mean again by that?
So basically, nature likes using simple parts, and it designs enzymes with a very precise function, and it designs thousands of those enzymes that can catalyze a particular reaction. And then it's just like pick and choose. You use those enzymes in a particular sequence, and then you get access to quite a diverse, structurally diverse molecule. So that's what I mean by modularity. It's not dissimilar from that robotic assembly line picture that I showed you. So by changing some of the robots in that pathway, you can build a Ferrari, you can build a Tesla, or you can build. Nature can build different molecules.
I have a question. So you told a little bit about history as well and how they used natural colorings. I was wondering, how do you know maybe how did they preserve those colors? How did they conserve them to keep them the same color?
So the stability of those colors, I don't know much about why they're fading with time. It is so basically because they absorb light, then this light may eventually destroy them. But I didn't look into how to preserve those colors. You probably would have to protect those molecules from the light by some extra layer, but. Yes?
The part where you showed us those molecules that protect against light damage, it seems like the pigments are all from one side of the spectrum. They're all in the reds and yellows, oranges and pinks. Is there a reason for that? Like, is there a certain kind of light that's more damaging? Or?
So I didn't have time to show you more pigment. The pigment that protects us as humans from light is melatonin, sorry. And it is a structurally completely different molecule than the carotenoids I showed you. Like we, as animals, as humans, are unable to make carotenoids. We have to get them from our diets. But our body is capable of producing that brown dye that gives us the suntan, that protects us from the sun damage. There are other colors like purples that other organisms use. I just didn't have enough time to cover them up.
So, is there a reason that despite the spectrum being sort of from red at the long wave end to violet at the short wave end, we perceive it as being more of a color wheel? So we see reds and purples as being adjacent somehow, right? Is there some biology behind that as to why we see them as being adjacent, or why there's such a color as red-purple?
I don't know the answer to that question because it's more of physics. I think the spectrum is linear, and it's just the superimposition of certain wavelengths that give us the cross section that you referred as circular. It's the spectral, the length of the wavelengths is definitely linear.
I actually know the answer.
What is that?
It bugged me for years why red and blue combined would look the same thing as something with a totally different wavelength. The cone that absorbs red light also has a smaller absorption peak in the purple end of the spectrum. And so that's why stimulating the red cone and the blue cone gives you the same experience as purple light, because purple also stimulates both.
Mm-hmm.
Cool.
Yay! Well, great, thank you. Let's go to Ania. Coming. Please stick around and enjoy beverages and food, and Ania will be free to answer some more questions. We have a mailing list over there if you'd like to sign up, and we also have some nice little postcards.